Hello class, this is Mr. Mormon. Welcome to video 5 in our genetics series. Today we'll be talking all about DNA. Let's start with the discovery of DNA and its important role in biology. Now, Mendel taught us a lot about inheritance. He showed that both mother and father in sexually reproducing species contribute to the genes that get passed on to the offspring. But nobody knew exactly what it was that was where were the genes? How did the genes work? What were they made of? And how did they determine the characteristics of the offspring? It was the work of many other scientists that proved that it was DNA was actually the molecule that was carrying the hereditary information. It started with the work of Frederic Griffith. Griffith was studying pneumonia. He was trying to figure out how the bacteria pneumonia caused sickness in people. He was doing work with mice. Now, Griffith had different strains of the pneumonia bacteria. One strain actually didn't make the mice sick. The virulent strain, when injected into the mice, would kill the mice because they would get pneumonia. When Griffith killed the bacteria by heating them up and injected them into mice, the mice didn't get sick. But what he found was when he combined the two, the heat-killed bacteria and the bacteria that couldn't get the mice sick, he actually ended up with the mice died. And when he looked inside the mice, he found living virulent bacteria. Somehow the harmless bacteria were transformed into the harmful ones when the dead cells were present. Something was being transferred that changed them. So Griffith knew that whatever it was that was transferring from the dead virulent bacteria to the non-virulent ones must be the genetic molecule, what was carrying the genetic information. He didn't know what it was, but he knew that must be it. Canadian biologist Oswald Avery took the experiments a little bit further. Avery made extracts from the heat-killed bacteria and then carefully treated the extracts with different enzymes to destroy different components in there to see what would happen. When he added the enzyme that kills or destroys RNA, the transformation still happened. So it wasn't RNA that was carrying the information. When he used enzymes that destroyed proteins, didn't happen. Lipids or carbohydrates, destroying those didn't make a difference. The only one that seemed to change things was when he destroyed the DNA. When he added DNA destroying enzymes, there was no transformation. So that proved that it was DNA that was carrying the information. Further proof came from the experiments of Hershey and Chase. What's interesting about these scientists is that they worked right here on Long Island at Cold Spring Harbor Laboratory. They did their research on bacteriophages, which are viruses that infect bacteria. And here we see the viruses infecting the bacteria. They added radioactive markers to try to find out what it actually is that the virus is transmitting to the bacteria, because they knew that was the genetic material. Now, a virus has a protein coat around the outside and DNA on the inside. They did different experiments where they added radioactive markers to the DNA or they added it to the protein. And what they found was that it was the DNA that was traveling into the bacteria and sticking with them, and it was the protein coats were being shed afterwards. So they could track what was happening, even though they couldn't see these tiny little particles, these viruses. They knew from the radiation that they had added to it that it was the DNA was the molecule that was carrying the genetic information. Now, discovery of the double helix shape of the DNA molecule, double helix meaning you have two of these twisted spirals making like a twisted ladder shape or a windy staircase shape, is credited to Watson and Crick. But credit also has to be given to Rosalind Franklin, who did a lot of the X-ray crystallography work that made Watson and Crick's discovery possible. Here's a famous picture of Watson and Crick standing next to a model that they were working on. They were chemists and they were working on the shape for a long time, but they weren't really getting anywhere until they got a big breakthrough when they happened to see this X-ray crystallography image that was created by Rosalind Franklin. Now, I don't know how to interpret X-ray crystallography, but Watson and Crick knew right away from this image that the shape that they were looking for was a double helix. Okay, here's some of the vocabulary we should know about DNA. Double helix, the twisted ladder shape of DNA, and the building blocks are called nucleotides. So in this model here, we see all these different building blocks. Each one of these is a nucleotide. Now, a nucleotide has three parts. There's a phosphate group and a sugar, a 5-carbon sugar here. This is deoxyribose in DNA. And then you have a nitrogenous base. In this case, we're looking at cysteine. That's the C. But there's 4 of them. It could be adenine, thymine, cysteine, or guanine. So the nitrogenous bases are the 4 letters that are used in the DNA alphabet, or the DNA code. A stands for adenine, T for thymine, C for cytosine, G for guanine. Now, there's a rule with how these nitrogenous bases pair together. A always goes with T, and G always goes with C. Now, the order of those nitrogenous bases along a gene forms a genetic code that specifies the order of the amino acids that make a protein. Now, the order that those amino acids get put into is very important because that's going to make the protein in a special shape. It will fold and bend depending on what order they are placed in, and that makes the protein in its special shape. And all proteins work and do their special jobs because of their special shape. Now, a single gene will contain anywhere from several hundred up to more than 1,000,000 base pairs. Now, you should understand that the code or message comes in sets of 3. It takes 3 nitrogenous bases to spell out the information for one amino acid. So these 3 nitrogenous bases could spell out the information for a specific amino acid. That would be a codon. Here we see some more diagrams that show what a codon is. Now, a change in DNA is called a mutation, and there's different types of mutations. In a substitution, one nitrogenous base gets replaced by another. And in addition, a new nitrogenous base gets squeezed in, and in a deletion, one gets left out. Now, there's different causes of mutations. Anything that can mess up DNA is a mutation. These are all things, by the way, that can lead to cancer as well. Radiation, chemicals, and infecting agents like viruses. Mutations that happen in body cells can lead to cancer. Mutations that happen in sex cells can be inherited and passed on to the next generation. And only the mutations of sex cells can be passed on. Now, most mutations are harmful, and therefore they end up, kind of dying out of the population. But every now and then, a mutation comes along that's beneficial, and that will become more and more common in the population. And this is how species change over time. It's the basis of evolution. So in summary, DNA was discovered to be the molecule of inheritance. It is the blueprint of life, containing the genetic information on how to make proteins. Its shape is a double helix. The rungs contain the genetic code. The A's, T's, G's, and C's spell out the genetic code, the information, which comes in sets of 3 called codons. Each of the building blocks is called a nucleotide and contains a phosphate group, a 5-carbon sugar called deoxyribose, and a nitrogenous base, either adenine, thymine, guanine, or cytosine. A's always go with T's, and G's always go with C's. Okay, this is the last thing I want to talk about, DNA replication. That's copying the DNA molecule. Now, there's many, many enzymes involved in this process. Two of the main ones that you should know about are DNA helicase, which is this enzyme right here. You can see that it's moving along and unzipping the DNA strands. The double-stranded DNA is being turned into two single strands. The DNA polymerase then binds and adds the proper complementary bases to build the new strands of DNA. What you end up with is two strands that are, two double-stranded DNA molecules that are identical to each other because of the rules that A has to go with T and G has to go with C. There's only one way that it can go back together, and you end up with two identical copies of the original DNA molecule. Okay, that's it for this video lesson. If you have any questions, as always, ask me tomorrow in class.